Welcome to Manifold. I'm very excited today to have as a guest Professor Shai Carmi of Hebrew University in Jerusalem. Shai is a distinguished and very well-known professor of statistical genetics and computational genetics. He also is a Twitter explainer of other people's scientific papers. So I think that's not the way I first became aware of Shai, but one of the ways in which he impinged heavily on my consciousness, because there's some set of people who are following the field really carefully, and when they see an interesting paper, they actually put out a Twitter thread on it or tweet on it. And my lab has benefited very much from Shai, because actually all the postdocs in my lab follow Shai's Twitter feed. And well, it's sad but true, but science is now kind of being centered on Twitter these days. Shai, welcome to Manifold.
Hello, welcome. Thank you.
So I would like to start with your early life and education. And I discovered, embarrassingly for me, not only just recently, that you also began your career as a physicist. And so you have undergraduate degrees in physics and computer science, and then your PhD was in physics. And I think it looks like you started in some kind of statistical or network kind of analysis, kind of physics. But then by the time you finish your PhD, you are already kind of getting into things like gene editing and things like this. Maybe you could just tell us a little bit about your intellectual journey.
Yeah, sure. So I studied at Bar Ilan University, that's in central Israel. And as an undergrad, I studied computer science and physics and a little bit of math. By the time I was in like third year as an undergrad, I already started research in a computational group in the physics department there. And I worked in statistical physics towards my master's degree and PhD. And the subjects that I worked on were network theory, as you mentioned. We studied transport processes over networks. In other words, random walks or search or navigation over random networks, and sometimes over real networks like the internet. Um, and I did that for a few years. And then I also studied diffusion equations, so-called anomalous diffusion theory. Um, and then by about 2007, 2008, I decided that I would like to try the life sciences. I would like to learn more biology and see if I can do research in that area. It really interested me. So I took some classes and I started talking to some people. I also worked in a wet lab for about a year, doing experiments with RNA, molecular genetics experiments. And then a year later, I even joined a lab still at Bar Ilan University in the life sciences, a lab that worked on RNA editing. So that's that's a few years before CRISPR and all that. We studied changes that happen to the sequence of RNA after transcription. So I did that for about another year. And then I moved to the United States for my postdoc. I did my postdoc at Columbia University in New York with Itzik Pe'er. The things that I studied there were population genetics and later medical genetics. And these are still the areas where I do research in my group at the Hebrew University. So I've been at the Hebrew University since 2015. It's about almost 7 years now. And I'm at the Faculty of Medicine at the School of Public Health. And I'm still working in population genetics. Actually, it's about I spend about half my time in that field, developing theory, methods, learning how to use genetic variation to learn about the demographic history of populations, about mutations and recombinations, about these processes. And specifically, we're interested in the demographic history of Jewish and other Israeli populations, and also recently in the context of ancient DNA. And in the second half of the activities in my lab, we work in medical genetics, and we have few collaborations studying specific diseases. But we also have some projects on screening, preconception, carrier screening, and also pre-implantation genetic testing. And I mean, maybe I can share the story of how I got to this to work on on subjects so diverse, from Ashkenazi Jewish genetics to pre-implantation genetic testing. In other words, testing of embryos. So in my postdoc, just about 10 years ago, we started a large sequencing project in the Ashkenazi Jewish population. So it was a consortium, about a dozen researchers from the New York area, and we sequenced nearly 130 whole genomes, which at the time was quite a lot, still quite nice. And I led the analysis of this data as a postdoc. We used this data to study the history of the population. For example, we studied a founder event in the history of Ashkenazi Jews. The population was very small until about 700 years ago, which led to a lot of disease mutations, for example. And we also did some work in medical genetics with this data. And for example, one of the applications of this data, one of the ways this data is used is for improving imputation. So if we have genetic data coming from microarrays, which is sparse and only has about 500,000 or so genetic markers, we can use whole genome sequencing data to impute, to fill in all the missing variants. But by finding similarity between the genome that we're trying to impute, the genome that for which we have missing data, similarity between that genome and the one of the genomes that we sequenced. By the time I was about to return to Israel, someone approached me from the Shaarei Tzedek hospital in Jerusalem, from the pre-implantation genetic testing lab there. And they were trying to improve the accuracy of methods for sequencing the genomes of embryos for specific disease mutations. And because many of their patients have Ashkenazi Jewish ancestry, they thought that by using these whole genome sequences, they could improve the accuracy of genotyping of the embryos, which is difficult because of because it's basically a single cell experiment. There is very little genetic material to work with. I started working with this person. His name is David Zevi. And then we published a little paper about these Ashkenazi reference genomes, but we also started developing methods more generally for sequencing the genomes of embryos using very low coverage sequencing of the embryos. And we published a couple papers on these methods, which have both an experimental and computational innovations. And we're still working together. And by the time I became more knowledgeable in how PGT works and what are the challenges in sequencing embryos, and just the whole procedure of how PGT works. I also learned about all the developments in the genetics of complex traits and diseases. I thought that now that we have whole genomes available for the embryos, which actually surprised me. It's we could achieve very accurate coverage of those embryos for basically just $100 per embryo. And then I thought, if we have all this information from the embryo, what else can we do with this information? And then all those studies started to come out with polygenic scores for predicting traits and disease risk and so on, which led me to start thinking about screening IVF embryos for polygenic traits and diseases. And since then, this is when I started this series of research projects on this subject.
Great. That's a great segue into I think we're going to discuss several of your papers if we have time. But it reminds me, we need to make a couple disclaimers. So I'm a co-founder of two companies, Othram, which does forensic DNA analysis, and another called Genomic Prediction, which does pre-implantation genetic screening of embryos. And you are in no way endorsing those companies. You are only stating your scientific opinions based on your own research. And we should not ascribe any endorsement to you for the work of those companies. Similarly, you are a consultant for MyHeritage DNA, which is I guess you could call it a consumer DNA company, which I guess is focused both on ancestry, maybe, and also on health risks.
Right.
And I guess we should just point that out for any conflict of interest reasons. Is that fair?
Thank you. Yes. Thank you very much for pointing this out.
Great. Now, I'm really interested in your paper. So you wrote a paper on analyzing the gain or the utility gain from embryo selection, I guess I should say risk reduction from embryo selection. And one of the phenotypes that you looked at was schizophrenia. And I have a personal connection to this because in high school, one of my close friends who was actually Ashkenazi Jewish American, but
Okay.
we were growing up in the middle of Iowa. His father was a history professor. He was a brilliant kid, and he went to Princeton to study. He was a double major in biology and English. But then he had an attack of schizophrenia in his early 20s, and he's never been honestly normal since. He's been a actual, I would honestly say kind of a burden to his family for his entire life because he never really recovered. And that was a very strong shock to me because intellectually this guy was very strong, very ambitious, and he was just struck down by this disease. And so I've always had a little bit of an interest in schiz. I don't know that much about it, but for this personal reason, I have an interest in it. My understanding is it affects something like 0.5% roughly. I mean, it varies from population to population, but
Even up to
half a
1%.
even 1%
Yeah.
of the general population. So
Yes.
it's not a super rare
Right.
condition, but it can be devastating,
Right.
not just for the individual, but for their whole family, as I understand.
Right.
And if you have a family history, conditional on having a family history of schizophrenia, the individual risk for just conditional risk might be as high as like 10%.
Hmm.
So in your analysis, you looked at what would happen if you could use a genetic predictor, polygenic predictor of schizophrenia risk, and you had some number of embryos. Let's say, I think was the number four. One of the cases that you looked at was
We
maybe four
mostly looked at
embryos.
5 embryos.
Five, sorry, five. So say you pick the lowest schizophrenia risk embryo out of five, you could have a relative risk reduction. I'll define in a minute what that
Hmm.
is. Relative risk reduction of something like 50%. Is that,
Yes.
was that your result? Okay.
Yeah.
Now, relative risk reduction means if the embryo, if there was some pre-existing risk for that family, then the reduction was 50%. So by going through this procedure, they would have half the amount of risk. However, the absolute reduction in risk maybe is only 1% down to 0.5% if the family doesn't have any history of the condition, the absolute risk reduction. Whereas if they had a family history, the absolute risk reduction might be much larger, like maybe from 10 to 5% or something like that. Is that a fair summary of
Uh,
your results?
yes, pretty much. Yeah. Would you like me to
Okay.
expand?
Yeah, absolutely. Please do.
Yeah. So fortunately, I mean, I didn't have any personal encounter with this disease. The interest in this, specifically in this disease, comes mostly from my colleague Todd Lance, who is working specifically in this field of psychiatric genetics. But also, I tend to believe that psychiatric diseases are probably going to be popular targets for polygenic embryo screening, either in companies like yours or elsewhere, because they can be so devastating. So to find the expected relative risk reduction, so yes, we assume that there are 5 embryos available. And then we use two approaches to compute the relative risk reduction. And in both approaches, we assume that the parents would sequence the genomes of all 5 embryos and compute the polygenic score for schizophrenia for each of those embryos. One important point is that we're assuming that the parents are considering only this single polygenic risk score. They're only considering this one disease, not testing for multiple diseases. This is something we can talk about later. And then the parents are going to be transferring the embryo that has the lowest risk score for schizophrenia. And then we use two approaches. The first is mathematical, a statistical model, which is called the liability threshold model, which states basically that each disease has so disease is a binary trait or a binary variable. An individual can be healthy or sick, but it is assumed that each such disease has nevertheless a continuous so-called liability that is unobserved. But this is something that we cannot measure it, but when the liability becomes very high, we see the disease. The person becomes infected. The disease appears. This happens to be a good model for fitting various types of genetic data. And the assumption is that this liability has a genetic component and a non-genetic or environmental component. And the relative sizes of the genetic and the non-genetic components are dictated by the heritability. But we don't even know the entire genetic component. We don't know every single gene, every single genetic risk factor for the disease, we only know the polygenic risk score, which is basically just the number of risk alleles that are carried by an individual based on results of genome-wide association studies. So these studies, they look at cases and controls. They try to find associations. They find typically hundreds of alleles that are associated with the disease. And then for a new individual, we just count how many risk alleles we have. It's a good idea to do a weighted average, to give a weight for each allele by the risk of carrying that allele. But these are technical details. Eventually, we assume that the liability has the genetic component represented by the polygenic risk score, and then a residual component which comprises all the other genetic risk factors plus environmental factors. And then, under the very reasonable assumption that these components are normally distributed, then we can compute the distribution of the polygenic score for the embryo that will be selected, and from that we can compute the risk of the embryo to be affected by the disease as an adult. And then we can compare it to the average risk in the population, and from that we can compute the relative risk reduction. That was the first approach. The second approach was to use real genomes from cases and controls. So we cannot do an experiment. There's no data we can do. There's no data we can get from an experiment where we would actively select an embryo based on one strategy or another. So we still have to rely on to some degree on simulation. So we take genomic data from cases and controls. In this case of schizophrenia, cases of schizophrenia and individuals unaffected with the disease. And then what we do is we call it virtual couples. So we mate individuals at random to create pairs of father and mother. Although they don't even have to be male and female, but we just create those pairs. And then we use, I would say, standard methods from genetics to simulate genomes of children. And then once we have the genomes of children for each such virtual couple, we can compute the polygenic scores of the children. And then we can compute the risk of the children to be affected based on models that were learned for the parents. So we use the parents, we use those case control genomes to learn what should be the risk of an individual based on the polygenic score. And then we use these models to compute the risk of each embryo, and we select each simulated embryo, and then we do this experiment, computational experiment, where we say, okay, what is going to be the risk of the embryo that has the lowest polygenic risk score for schizophrenia? And then we compare it to selecting a random embryo, equivalently to the prevalence of the disease in the population. And again, from that, we can compute the relative risk reduction. And what we found is that for schizophrenia, the polygenic risk scores that we have right now, they, in technical terms, they explain about 7% of the liability, of the variance in liability of the disease. It's a way to quantify, to measure the predictive power of the polygenic score, which is not very high, but it's also not very low. It's about typical for many diseases. Most diseases today have polygenic scores explaining the variance of about 5 to 10% of the liability. And given this parameter, and given that we have 5 embryos for each couple, then our estimates were that the relative risk reduction is going to be between 40 and 50%. As you mentioned, this is the relative risk reduction. It's not the absolute risk reduction. So if the disease has prevalence of 1%, this means that the absolute risk reduction is going to be only 0.5%, or more correctly, 0.5 percentage points. And this is something that we need to be aware of, that risk reduction that may seem very large as a relative risk reduction is actually quite small on the absolute scale. And both ways of presenting the risk are correct, but I think it's usually better to present both to make sure patients know what to expect. The last point that I'll mention, again, as you said, like what happens when one of the parents one or both parents are affected, and of course it increases substantially the risk of the child. So as you said, I think these are about the right order of magnitude. The children of an individual with schizophrenia have probability about 10 times higher to become affected compared to random individuals. So it's about 10% prevalence. And what we showed is that the relative risk reduction is still remains, I think it's if I remember correctly from the the numbers we reported, I think it's a little bit less, maybe not 50%, maybe closer to 40%, but it's still quite high. And the important thing here is that it's also relatively high on the absolute scale. So risk reduction can be 4 percentage points, which is perhaps more meaningful. I thought that these derivations or this part of the modeling that we did is quite important because these might be the first use cases for polygenic embryo screening, perhaps not in the US, but in other countries, these might be cases where parents will ask for their embryos to be screened or prioritized based on polygenic scores in case one of them is affected by schizophrenia or similar diseases.
Yeah, on that last point at Genomic Prediction, a lot of the parents coming in who request our embryo reports, they have a family history of some pretty impactful condition like breast cancer. I don't know if we've had schizophrenia yet, but breast cancer and heart attack are two of the big ones where if there's a family history, then the family is aware of that. And if they're not carriers of BRCA or some other Mendelian variant, they know it's polygenic in nature, and then they're interested in selecting an embryo which is lower risk. I wanted to ask you a technical question. So we did some complementary calculations where it turns out in UK Biobank and in other data sets, there are siblings. And so we were able to look at siblings who, their average age is in the 60s or something. So they've already lived, big chunk of their lives. And then you can ask how well do the predictors allow you to predict which of the two siblings has the condition and which one doesn't. And for more common conditions, you can easily assemble enough data to figure out, say, how well the heart attack, CAD predictor works, or hypertension, or whatever it is. And it's interesting because those are based on real people, but we get very similar results to what I think you and other groups got from a little more simulated kind of analysis. So that was quite interesting at a scientific level. I had a very technical question I wanted to ask because we've thought about making synthetic babies, not I mean, on the computer in silico. So the question I always have, and this is, exposing my lack of biology knowledge as a dumb physicist, is when you have meiosis and you make gametes, so you make an egg from the mother, you make a sperm from the father, we sort of know the chunk sizes that, the gamete only gets half DNA of say, the father. Do we know much about the distribution of how those chunks are determined in meiosis? Like, I would think this is something that you could actually answer this question through data now.
Yeah, yeah, we know. We have a pretty good idea. We have something called the genetic map, which basically for every position in the genome or for every small window of the genome, it provides the probability of having recombination, crossover, in that window in each meiosis, in each generation. So with these genetic maps, we can easily model meiosis. That's exactly how we implemented our simulations. And these ideas are also very commonly used when people are trying to study, for example, methods for detecting relatedness. So this is like if I have a new method to detect relatives, then I would need to simulate these relatives, and this is how it's done. I would have the two chromosomes from each parent, and then I need to draw the positions of crossovers. And the standard assumption is that the position is uniform along the genome. It's not exactly accurate. Also, separately, I wrote a different paper about when this assumption does not exactly hold, but broadly, it's fine to make this assumption, and simply it's a Poisson process. If you go even more technical, and the rate of the process is simply determined by the total so-called genetic map of the chromosome. So this is how we drew the chromosomes. The one problem with this method is that we don't really have we don't really have the chromosome sequence, the haplotypes of an individual. We can only infer them computationally using something called phasing, which is imperfect. So in a sense, like each of the chromosomes that we think we have for an individual is actually a mixture of the two chromosomes of the individual, but at least over relatively short range, something like 100 kb or 1 megabase, we have phasing is quite accurate. So I think what's important is that we get the local LD structure, we get it right in the children. But otherwise, the precise positions of the crossovers, it doesn't matter that much.
I see. Wait, so as a first approximation, you can take it to be just a uniform probability at each position for
Yeah,
uh,
there is something called crossover
recombination.
interference, which means that not only in humans, in many organisms, there is two crossovers cannot be too close together. There's like a repulsion between crossovers, and it can be modeled, but overall, it's not very important. I think in this for the purposes of studying, risk reduction.
Yeah, I mean, if the predictor is super polygenic, this will kind of come out in the wash, I guess.
Yes, yeah,
Um,
exactly.
the the sort of basic biology question that I was ignorant of is I thought I had heard that there are quote recombination hotspots where the probability
Mhm.
just shoots up.
Yeah,
And the question is whether you have to model that
yeah.
in your
Right.
yeah.
So we did, as I explained, like we have this genetic map, which is in windows that are hotspots, there is higher probability. So maybe just to correct one thing I said before, we select the position of the crossover uniformly, but in genetic map units. Yeah, and that takes hotspots into account.
Got it. Yeah, so that's a very beautiful work and extremely useful, right? Because if you can generate synthetic siblings or offspring, right, there are many uses of that. And you actually hinted at another one, which is genetic genealogy and DNA forensics, which maybe hopefully we'll get to that later in our discussion. Um,
Yeah, I can also say that in terms of this, working with siblings, I think it's a great approach because it avoids some of the difficulties that we have with simulation. So I think the genomes of the simulated embryos that we generate on the computer, I think they are pretty realistic, but the problem is we still have to rely on a model for the risk given the polygenic score. And by working with actual siblings from the UK Biobank or other data sets, this problem is avoided. So it's a lot more realistic. The problem is working with siblings, it's more difficult to find out what happens with more than two siblings. So maybe I'll just take the opportunity to tell about our previous study where we looked at screening embryos for height, where we did a similar experiment, but we had a really unique and interesting data set. We had about a data set of about 400 Ashkenazi Jews from families, from very large nuclear families that in these families we had both parents, and the number of children was anywhere between 3 and 20. The average number of children per family was 10, and they were all
are these
adults, so we knew their height.
orthodox? Are these orthodox families?
Yes, exactly. They were Jewish Orthodox indeed. So and this community, in this community typically there are very large families, and that was quite nice because we could study empirically what happens if you select the embryo. I mean, it's not really an embryo; it's the child. It was a thought experiment. What if these children were embryo? I mean, it's the same thought experiment as you did with the UK Biobank, but we could go into very large numbers of children. But we only had height. It was only a study of height, unfortunately.
Yeah, I wasn't aware of that. So thanks for bringing that up. So I'm curious, first of all, the the height predictor, if it's trained on sort of a general European population, do you know what the fall off in variance accounted for or correlation is when you use it on Ashkenazi Jews?
So it's not too bad. I mean, from all of our experiments in working with polygenic risk scores or polygenic scores in general in Ashkenazi population, performance is quite similar to the European population. Actually, recently, just a month ago, we published a paper. I was a minor author on that paper. It was led by Florian Privet and Bjarne Wilhelmsson from Aarhus. They looked at the UK Biobank and studied the transferability of or the relative performance of polygenic scores across populations from within the UK Biobank. So there isn't a problem of using different
Arrays.
arrays, exactly genotyping platforms and so on. And we found that so relative to Europeans, in the Ashkenazi population, the performance of the polygenic scores is 85%. So there is like a 15 percentage point decline in the performance of the scores. which is not too large. So these scores work pretty well. Even the schizophrenia study in the newer paper on diseases is also from the Ashkenazi population, and performance was quite good.
Yes, that's great. This large family idea is excellent because I often find people have the wrong intuition about how much variance is present amongst their children. So some people think there's no variance among children, which is very strange, right? Because I remember in high school, there were families where one brother was, 6 inches taller. That's a little bit rare, but 4 or 5 inches taller than the other brother, and so it immediately tells you. Or one was a really good student, and one had a lot of trouble at school. So intuitively, I think if you're a careful observer, you know that within a family, the kids can really differ. Or if you're a parent, even the kids can differ a lot. But I think obviously you can quantify this, right? So it's sort of half the variance in the among SIBs as in the general
Yes, I mean, I agree that it's not obvious intuitively. I mean, maybe even if you had asked me like 5 years ago or 10 years ago what should be the variance within the population, then I wouldn't have guessed half. But it's a classic result in quantitative genetics that variance within siblings is half the variance in the population. And I think this is relating to the statement by the European Society of Human Genetics. I mean, that was, I think, one of the main issues where I had disagreement there. But it's kind of typical to a lot of the criticism that I've heard about polygenic embryo screening. So I mean, a lot of criticism is maybe justified and worth, or at least worth discussing. But that point, I think, is just I mean, you just have to be familiar with this mathematical results, and the claim that there is not enough variance, I think, is simply it does not hold. That said, the fact that there is half the variance, as we showed in that previous paper on traits, it doesn't mean the gains are going to be particularly high. For example, in that paper, we showed that for IQ, in that paper, we even talked about selecting out of 10 embryos, the average gain in IQ was only around 2 points or 3 IQ points, which is, I think almost everyone will agree is very little. But for diseases, turns out that the impact can be quite large.
Yeah, I should point out that our company only does health-related risks. We don't do IQ or anything like that, or height. So you mentioned this ESHG, so that's European Society of Human Genetics, and they wrote a viewpoint, I guess, which was kind of negative about embryo screening. And I guess my attitude is like yours. If somebody has a valid scientific point, then I want to understand it as much as I can. I have a responsibility to understand it as much as I can and discuss it with them. But then if they have some argument like, "Oh, there's no variance among SIBs, that's why this is not even worthwhile doing," it seems kind of wrong-headed. And you wrote a very nice, I think it was kind of like a one-pager that you put on Twitter, which really dissected their statement. And I think I agreed with you on every single point. Now, to me, the really valid issues that they raise obviously are things like, well, we don't have the predictors, as we were just discussing. They work best in the populations where we have the most training data. And the more distant you are from those, say, European populations, the less well the predictors work. That's the current situation. And that's a problem the whole field has to fix, not just anybody involved in IVF or embryo testing. And the other issue, I think, is that there has to be I think you said this as well, there has to be a big society wide discussion of how this should be regulated, what should be allowed, what should not be allowed. Of course, it should be based on the best science, right? So it shouldn't be people who aren't familiar with all the recent research giving their opinion. It should be an informed opinion.
Correct. So, I mean, there was some criticism I agreed with, and we also raised some of the points in the comment that I wrote with colleagues, bioethicist colleagues. But yes, I mean, some data is out there on the expected outcome, so no reason not to use it. And of course, I mean, I think everyone is aware of the issue with the lower performance in non-European in populations, or individuals with non-European ancestry. I think there is another issue with unmixed individuals that I think was not mentioned or was not discussed enough. I hope in our group we'll study it in the near future, and maybe it's a good opportunity to mention this. is that what happens if we have an admixed individuals, like if the couple, if one or both individuals of the couple are admixed, and for example, African Americans or Hispanic in the context of the United States, then the problem is that what happens is that the embryos will vary in the proportion of European ancestry that they have, because individuals themselves, they will have ancestry partly European and partly other, and for example, African. And then because of the randomness of meiosis, so-called Mendelian segregation, some embryos will have more European ancestry than others. And then the problem with polygenic scores, actually there are two problems associated with non-European populations. One is lower predictive accuracy, and the other is bias. So the polygenic risk scores at least tend to be lower, reflect lower risk in European populations. And if we are going to be selecting an embryo based on having the lowest polygenic risk scores, what may happen is that we may simply be selecting for more European ancestry. And this has the obvious social problem, but also it means the risk reduction that may be achieved may not be as high as we expect in Europeans. That's a bit more subtle, I would say, problem with implementing polygenic embryo screening in the context of admixed individuals, but I think it's worth thinking about as well.
Yeah, I think it's a very serious problem. And I think one of the interesting ideas that we've discussed is if you can do block by block, haploblock by haploblock identification of, which ancestry group that chunk of DNA came from, then you might apply a specific predictor. So one trained in Africans on certain subblocks and then one trained on Europeans on certain other subblocks and get some kind of aggregate score. But of course, this requires a lot of research to really be able to do it. I think another thing that I guess this is a broader issue, but one of the things that I think the ESHG letter or viewpoint said was that there's no way to know whether any of this, maybe I hope I'm not straw manning what they said, but it seemed like they said you can't know whether any of this ever works until lots of these babies grow up. And, you decide, did the polygenic risk really correspond to the actual risk for that set of kids? That seems like a very unrealistically high standard. And I'm curious what you think about that. I think you commented on that.
Right. Yeah. So I think they are aware that it's infeasible to do these experiments. So let me just reiterate what they were saying. They were saying that if we have this possible intervention of prioritizing the embryos based on polygenic scores and then transferring based on these scores, if you want to do this intervention, show that it has health benefits, we need to do, let's say, randomized clinical trial where we would, randomize some couples to select embryos by polygenic scores and others to select embryos at random or based on morphology and so on. And then wait, a few decades until there are health outcomes and then see if this method worked to reduce the disease burden. So what they said is that it would be impossible or irresponsible to use polygenic embryo screening before such trials are conducted. So I think they are well aware that it is impossible to conduct such trials. I don't think they seriously suggested to do them. But I think their point was that it would be, because it would be impossible to run such trials, we should be careful. I don't know if ban, but we should be very careful with using polygenic scores with embryos. But I think that we have the tool of statistics and we can develop models or even simulations like based on the UK Biobank, as you did. And I think these tools can give us reasonable estimates regarding what we expect. And I think what is important is not the result of one specific model, one or another model, or one or another paper, but the triangulation of evidence. If we have evidence coming from multiple methods, multiple approaches, distinct approaches, and distinct groups that shows similar results, then I think we can trust these results to some extent. At least get started with implementing this screening by polygenic scores.
Now, the analogy that pops into my head, well, I guess there are two. One is that we already do, or it is fairly common to do Mendelian screening against rare Mendelian variants. And sometimes in those cases also, it's not fully understood any kind of causal biological mechanism by how the mutation is actually causing the disease, but it's just a statistical association between the presence of that variant and later development of the disease.
Yeah.
Um,
But I would say that for most, I think the difference is that for most Mendelian mutations, I think for most of them we understand the cause, or at least the evidence is overwhelming. I mean, I find it difficult to find. I mean, how many diseases we have for which we don't understand the biology of how the mutation causes the disease.
okay, I'm not an expert on this, but to me, it sounds like I guess being kind of skeptical always about biomedicine a little bit is that sometimes I go to a talk and they're explaining me what the mechanism is, and it looks like they just draw a cartoon, and then really they're appealing to the statistics to justify their belief in this. But the mechanism sometimes, I mean, obviously, if they have some intervention or some really very definitive biochemical uh, tests that can support the hypothesis, sometimes it seems like they're confident, but they should maybe the main source of the confidence to me seems like it's statistical data.
Yeah, I mean, in some cases, yeah, I don't know enough to, to say how widespread this is, but but I think it's not. At least in my opinion, I don't think the fact that we only have statistical evaluations of the expected outcomes. I don't think it should. I'm not saying we should approve the technology because of one statistical evaluation or another, but I think we should definitely consider. I mean, simply because we cannot wait for trials. I mean, this is clear, I think, for everyone that it is impractical to do these trials and to wait for the outcome. So we will have to decide whether it should be approved or not based on what we have right now, based on the tools that we have right now.
Right. And again, I think it's not that different from, when the FDA has to approve a particular drug. When I look at the analysis, I was some years ago, my doctor, I had a high cholesterol readout, which was anomalous, but he immediately said I should start taking statins. So I started looking into statins, and at the time, the data supporting the efficacy of statins seemed pretty weak to me, but it had been approved. It was already starting to be $1 billion business or something. And this Alzheimer's drug, I think it's called Aduhelm, that also seems to have been approved on fairly weak statistical data. So I feel like the evidence in our papers is much stronger than that. It seems like to me, but I'd like to hear your opinion.
I don't think I know enough to, to give a confident assessment of these other drugs, but I think, you know, particularly not on, Alzheimer's drug. The one difference is that for these drugs, or, in general, let's talk generally, not about one drug or another. In general, for drugs, there have to be clinical, like randomized clinical trials. So you measure the efficacy, you eventually you have to run a statistical test and to get a p-value, but you have outcomes from people who are treated compared to people who are not treated. And this is something that we will not have for polygenic embryo screening in the, not in the near future and also not in the distant future. And I think that's, in my opinion, that's a big difference. But whether that's a, difference big enough to justify banning polygenic embryo screening just based on that, probably not, in my opinion.
Yeah, I agree. I mean, that is a fundamental distinction. I guess when I was comparing to these other sort of tests of statistical tests for drugs, I was thinking about the delicacy of the statistical analysis and how, you have to make some assumptions in the analysis. And for again, not to get into a specific one, but I looked into this because I was interested. The Alzheimer's one, Aduhelm, there's negative risk too. If you take the drug, there's some bad things that can happen to you. And so you have to trust their analysis to decide the net utility is positive for people taking this drug. So,
That's
so
true. I
there
mean,
is
you have
some,
to, yeah. I mean, I agree. Sometimes the statistics can become tricky, but sometimes you have cases like the COVID vaccines, for example, where you don't really need
yes,
statistics to see the
right,
effect, right?
right. Now, another criticism often against this kind of thing, against embryo selection, comes from something called plyotropy. And I don't recall where the ESHG viewpoint mentioned plyotropy, but we looked at this pretty carefully, and as far as we can tell, for the major polygenic predictors for the major diseases, there's actually fairly modest pleiotropy, and in fact, if you select on an index, so this is something that the doctors immediately demanded that there be some kind of general health index that they could use because it's just simpler to think about for them and simpler for them to explain to patients. That if you select on an index, generally doesn't seem to be a zero sum thing going on that you can actually improve. You can lower risks across a bunch of different risks. And this seems to be supported by other people who independently are studying things like longevity and such that you can build a kind of polygenic longevity predictor. I'm curious what you think about this or if you've studied this at all.
So we studied this question in both papers, in the 2019 paper on traits and the recent paper on diseases. In the 2019 paper on traits, we found that if selection is done for, so it's a bit tricky. The gain per trait, it decreases with the number of traits that are considered. So what we showed in the paper that the gain that you have in the traits of the future children decreases with the number of traits that you're selecting for, and it's inversely proportional to the square root of the number of traits. So if you're selecting for 4 traits, the gain per traits will be only half what you get had you selected for only 1 trait. So, and if you're selecting for 16 traits, you will get only quarter of the gain. But on the other end, you're going to be getting gains for more traits. So like the net benefit is positive. So you will gain more by selecting for more traits, and I assume for more diseases. But the gain per each individual trait or individual disease is going to be smaller.
Right.
Which I think once you think about it, once you think of it, I think it's also intuitively clear that this is what should happen. Um, so the thing is with diseases, I never studied it myself, but from what I've seen, I mean, there is correlation between diseases, genetic correlation between diseases. But the thing is that usually this correlation is positive. Usually it's not very strong. I mean, of course, except for diseases in the same, very similar diseases like schizophrenia and bipolar disorders, of course, there is very high genetic correlation. But in general, if you across all domains of medicine, the correlation is usually not large and it's positive. So if you're reducing the risk of one disease, you will be reducing the risk of another disease. So I tend to agree that once there is like an index of like a health index of some sort, either based on longevity or based on a combination of diseases, it should probably reduce the risk of most diseases simultaneously. Although, as I said before, the risk reduction is going to be much smaller than like the 50% that I mentioned earlier when you're selecting for a single disease. But I think what we have to be careful about is specific pairs of diseases that have very strong anti-correlation, because this can happen sometimes. It's not very common, but if a couple is about to select for one specific disease that has high anti-correlation, strong negative correlation with another disease, then this should be taken into account, because our results from the 2021 paper showed that this can increase quite substantially the risk of the correlated disease. So I would say, like, to summarize broadly, I don't think it should be a major concern, again, because of the mostly positive genetic correlation between diseases. But I think this is something that everyone should have in mind, for some specialized cases.
That was a very nice summary. So if you just assumed uncorrelated risks across the different diseases, then this 1/square root of n, and n scaling that I think that you gave is very plausible, right? It's what you would guess. And if you look at net consequence, you have sort of n gains and 1/√n reduction. So you get root n gains. And then I think as a kind of caricature, we find the same thing that, generally what you find is weakly positive correlations. And so weakly positive actually helps you. So you get, I think,
Mhm.
very roughly, like the way a physicist would say it is, you're going to do a little bit better than root n gains.
Yes.
And
Uh, right.
yeah,
Right. But again, you have to be careful about specific cases, like specific types of diseases that can have very strongly negatively correlated other disease.
yes. The way I think I can describe the way that often the clinics and the parents are using these reports that we generate is that the doctor, honestly, this is maybe a flaw in the American system because the doctors are always, unfortunately, they have kind of a motive where they're just trying to do things as simply and quickly as they can. Generally, they like kind of having a overall health score. But then the parents, especially if they have a family history or something, they're looking specifically at a line item risk. Like, okay, I understand the overall ranking using an index, but then I want to see like what is going on specifically with heart attack because a lot of people in my family, had heart attacks or something. So that seems like a reasonably rational way for people to use this information. It doesn't seem like people are having trouble making use of it.
Yeah. Although I would be careful in that case in that setting, about the expected relative risk reductions if the identity of the selected embryo is eventually not the one with the lowest or highest health index. Maybe it could change the calculations to some degree. I don't know.
Yeah, it's very
Yeah.
subtle. I mean, the genetic counselors that we train takes a while for them to explain, triple R and ARR
Mhm.
to the patients, but
Yeah.
it's the real focus of trying to give them a clear picture of what the information says.
Yeah, your experience would be very interesting. I assume you will publish it, at some point, all the data that you have on the patient experience. These are indeed the kinds of concerns that we raise. That maybe negative sides with polygenic embryo screening, like we referred in some places to choice overload, just having too much information to comprehend, and just having to select out of a few embryos may make it difficult for parents, and also difficulties in explaining the probabilities, because the risk is probabilistic. It's not deterministic as the case of cystic fibrosis or similar diseases, and also explaining the distinction between relative risk reduction and absolute risk reduction. I think all these points are there are causes of concern, in the greater reproductive medicine community about this technology, and any data that you have on this, I think would be very welcome by everyone studying this.
Yeah, I agree completely on the importance of these issues, and I'm pretty sure the company will publish some results on all this stuff. I'm more involved just on the really in the weeds computational stuff, not that sort of thing. I probably wouldn't even be a co-author on that paper, but I'm pretty sure they will. This is Steve, adding some information in after editing. I checked with my colleagues on Shai's question about follow-up studies of our patients. Genomic Prediction is preparing more than one paper on this kind of data from patients consenting to participate in research, which is most of them. The studies explore patient experiences and attitudes toward polygenic screening and gather not only longitudinal health outcome data, but also the psychological outcomes of screening, whether the patient found PGTP useful, whether it induced anxiety, relief, or regret. Also, whether reducing risk of mental health disorders in their family gave them courage to have children. Also, whether their family and friends approved, or whether the patient elected to keep the testing a secret from their peers or even from their own child. Or whether they decided not to receive PGTP results at all, and if so, why not? So far, more than half said yes when PGTP results were offered. All very interesting stuff, which the company looks forward to publishing. Some of it has already been presented at an IVF science conference called ASRM, American Society of Reproductive Medicine, last year. You can find more information about the study designs at lifeview.com, which is our website, and also on the public pre-registration of the studies at clinicaltrials.gov. They have all been IRB approved and submitted before initiation in accordance with best scientific practices. Thanks. I wanted to switch. Okay, well, there are two. We're almost at it. We're at an hour now. Sorry, and there are two more things I want to discuss. If you can just give me a number, maybe another 10 minutes, that would be
Sure.
great. Okay, so one is genetic genealogy and solving crimes, because you wrote one of the early papers on this, which I think at the time I read and was influential in my thinking, because I had been making kind of back of the envelope estimates, which were with conclusions similar to what you got. And then the other thing I want to discuss was the Israeli health system and whether you think there's a good chance that polygenic scores for adults might make an appearance in, I think it's a single payer system. Is that right?
I'll explain once we get there.
Okay.
So,
So let's do 5 or 10 minutes on genetic genealogy, and then however much time you can spare on healthcare. So this other company I'm involved in called Othram now is, I think the CEO tells me, solving several really prominent cold cases a week. So it's like,
oh well,
these
okay.
are like cases that have been outstanding for, on time scale of decade or maybe sometimes multiple decades. And, they're able to, I think they've solved cases with as little as 15 cells equivalent of DNA. And so they're getting really good at dealing with contaminated, messy DNA in crime scenes and getting a fair amount of case volume from police departments, FBI, stuff like that. So it's a real thing. I think it will change law enforcement quite a bit. And again, I want to give you credit. And I forgot your collaborator's name. He's a computer scientist at Columbia.
Yeah. So the person who led that paper in 2018 is Yaniv Ehrlich.
Yes, Yaniv.
Um,
So
yeah,
I heard him give his talk at ASHG. I think it might have been 2018 or
right.
2019.
2018, just after we published the paper.
Yeah.
Yeah, he was at Columbia, in same department where I was in computer science, and then in 2017, he moved back to Israel, and he was the CSO of MyHeritage. And I'm a consultant at MyHeritage, as I mentioned, so we worked together on this paper. It was mostly led by Yaniv, and we also used data by MyHeritage to generate those projections for the ability to identify at least Americans based on their DNA in this genetic genealogy approach.
Right. So as I was saying, I saw this as an opportunity to solve crimes and do things. And I was making much simpler estimates. And so when your paper came out, I was like, wow, this is great. And so I think one result you
Yeah.
had was,
Yeah,
let's see,
I will
what
also
is it?
mention a paper by Doc Adge and Graham Coop. They also generated some estimates of the expected number of people that we will be able to identify using this method. Comes
Yes.
in parallel to us. Yeah, also
Right.
reaching similar conclusions. Yes.
So I think roughly speaking, you were saying if you have roughly 1 million people in your data set, and let's say we're just talking about Europeans, I think you said 60% chance of finding like a third cousin match.
Yes,
Is that
that
correct?
was about, yeah, those were about the
Yeah.
numbers. Yeah,
Yeah.
it's all
Well,
very sensitive to like modeling assumptions, but that's the order of magnitude. Once the databases become large enough to have 1%, 2% of the population, then it becomes highly probable that everyone will have a relative, at least third cousin or closer, in the database.
yep. Now, there's been a whole evolution where a lot of the initial data used for this came from kind of genealogy enthusiast websites where people had uploaded their genomes. And then there was some controversy about whether some people were willing to allow their data to be used to catch criminals, et cetera. And so the
Mhm.
whole thing has evolved in a kind of complicated
Yes,
way. And now
very
awesome.
rapidly. Yeah,
Yeah.
it was quite
And now
amazing how fast things developed. Yeah.
incredible. And actually at the science level, it's under the results. Like Othram knows a lot now about sort of the corrections to the first order kind of analysis that you and Graham and these other guys did, because they see much more population structure, right? And population structure even among people that are more likely to commit crimes, or there's a bunch of science that they could do if they were not so busy building the company. They could publish a bunch of papers on this kind of thing. But I'm curious what you think will happen. So one of the hypotheses I had at the very beginning when I was talking to venture capitalists and looking for a CEO to run the company and saying we should really start a company to do this, I thought that there would eventually be legal challenges that would just force Ancestry and 23andMe and MyHeritage to, allow their data to be searchable by law enforcement, because a very similar thing happened at the beginning of the internet where the police would always want to go to an ISP or some free email provider and say, hey, there's a crime being committed and we need to look at all these emails. And initially those companies resisted, but then the police got court orders and things. And now we've flipped completely to where all of those internet companies have compliance departments whose only job is to basically comply with law enforcement to dig up the data that the FBI wants or something. So I'm wondering what you think will happen in the consumer DNA business in the long run regarding this kind of thing.
Yeah. Um, Yeah, I mean, let me first just comment about like the development, like scientific developments in the field very quickly. I think that you commented correctly that there were not too many developments, at least on the question of like how to find the relative or how many relatives we expect to find. I think most of the challenges come from extracting DNA from sometimes highly degraded samples, sometimes even almost ancient samples, can be decades old samples. Sometimes there are mixtures and so all those complications, and then sequence these DNA samples to high quality. I think these are major challenges that are still not fully resolved, and I think there can be a lot of progress there that will make applying this method easier. And I think that there are also challenges in the genealogy part. So once you have your second cousin of the criminal or the victim, sometimes these are unidentified victims. Once you have them, once you have that second cousin, it's not always easy to find the person you're looking for. And I don't think there are like I don't think it's like there's been like major development in methodology that would help genealogists to do this work. I think it's still kind of a lot of cumbersome manual work that has to be done. But the question is like of how many relatives we will detect. I don't think this would change too much. I think now that databases keep growing, at some point it will be just everyone is going to be nearly everyone is going to have a relative. So this question is going to have less importance. Um, the your question is about legal aspects. I'm less familiar with this, but I'm also equally impressed as you that there were no court orders. I think there was only one case, if I remember correctly. But by and large, the companies that did not want to cooperate with the law enforcement on this, they didn't do it. I mean, MyHeritage, again, where I'm consultant, notably 23andMe and Ancestry, that have the largest databases, they were very successful, very aggressive in protecting the privacy of their consumers, but also not helping law enforcement. It's also a question what's the right thing to do. It's not obvious, but the decision of this company was to protect as fiercely as they can the privacy of their users. And yeah, it is quite interesting that it's successful. I mean, it is not practiced in Israel, this method, for various reasons. It's a bit complex. I won't get into it. But in the US, it's the forensic genetic genealogy is now very widespread. But I think the law enforcement agencies they now simply they just live with what they are allowed to to use. I mean, with GEDmatch and family tree DNA, and they probably gave up. Maybe there are some things happening behind the scenes that I'm not aware of,
Yeah,
but
there are
maybe they
things
just
here.
realized they just won't have that court order, so they just should not waste their time on it.
My description of what's happening in the US, I guess I have some special information, but I don't know how much of it I should share. But there is a lot going on in the background with proprietary data, building proprietary data sets. And the police are just happy because this is already, even in the current situation, a big quantum leap over what they could do years ago. The local incentive structure for the detectives and prosecutors is they're just trying to clear as many cases as they can. And for them to engage in some protracted legal struggle with, say, 23andMe, they don't have a good incentive for investing that much of their time and energy to do it. But it could happen easily if, like, some kind of politically ambitious district attorney or somebody wanted to really push it in, maybe in some red state in the United States. That's the fluctuation that I'm waiting to see. I think that fluctuation will eventually happen. But right now, the police departments are just happy that they can clear a bunch of old cold cases, that they had never had any hope of solving.
I think also they have. I mean, I assume they don't solve every case using the genealogy. They don't always find relatives, but even if they find relatives in half the cases they're studying, they probably have enough work to for a few years forward, to keep them busy
That
working on
is
those
exact
cases.
that is
Uh,
exactly right. Yes.
yeah, in particular, there are so many rape kits, for example, that from what I read, that were not even genotyped. There's so much potential to use it, to use this technology even before going to court with 23andMe.
Yeah, the rape kit issue is a terrible one because there are many, many rape kits that have not been processed, and it's also true that a very small number of offenders are responsible for a lot of the rapes. So there's a kind of some, I don't know if it's a power law, but you'll like this from your earlier work, but there's some kind of distribution where a relatively small number of people are responsible for a hugely disproportionate number of crimes, and so when you lock them up, it has very large kind of network effects, or not network effects, but disproportionately good effects. Let me switch gears because again, I'm conscious of the time and I don't want to keep you too long. So I'm curious about the Israeli healthcare system, and I just have the feeling that, well, I guess maybe at the crudest level, people are smart in Israel. So I'm wondering if it's possible they'll be the first to start using PRS constructively for adult healthcare.
Okay, so briefly, the Israeli system is so there is universal healthcare, but it's not, there's no single provider or insurer. There are four providers for HMOs. The largest is called the Clalit. It insures about half the population, and they recently became very famous after they published several papers on the COVID vaccine because they have really, really good, high quality longitudinal electronic healthcare data spanning almost 30 years, I think. They also have a really good research institution. And then there is another provider called Maccabi, which covers about a quarter of the population. And then there are two smaller providers. But every person in Israel, every citizen must be a member of one of these HMOs. So regarding polygenic risk scores, they are not being implemented clinically right now anywhere in Israel. As I mentioned earlier in this conversation, only very recently we have good data on the predictive power of polygenic scores in the Ashkenazi population, and we know that the polygenic scores work pretty well. Not exactly the same accuracy as in Europeans, but almost there. So we know that they should work well in Israel. Regarding the other populations of Israel, we know very little, if at all. I mean, I did like a very, very small study of 100 people from an Arab village studying LDL cholesterol, and that's it pretty much, I think. So there is a lot of, we don't know enough about how well polygenic scores are going to work. And we also don't know enough about the attitudes of people towards using polygenic scores clinically. And I'm trying to run at the moment, few small studies in context of cardiology and oncology to just take a sample of cases and controls from clinics in Israel and compare polygenic risk scores. And so we know it should work in Ashkenazi Jews. That we should validate that first, and then see how it works on the other, in the other populations, particularly the non-Jewish population. So we have this missing data. What we do have, which I hope will take off, in the near future, is a large national project to set up a biobank or a study of 100,000 Israelis, along with their genomes and their clinical records. And this project, it has a lot of funding secured from the government, but it ran into some barriers and technical and legal problems throughout the past few years. Now there's new management and I would say more excitement about it, and things are starting to move on. And I hope that this momentum will continue, and then we will have a really large resource of genomic data from Israel. And then, of course, with these amazing longitudinal medical records that we have in Israel associated with that we will have associated with these genomes, I think it will be very easy to study the utility of polygenic scores. And then I think there will be openness to try polygenic risk scores in the clinic. I think it will be important to have some preliminary cost effectiveness analysis or preliminary trials. But from conversations that I've had with clinicians, maybe it's a biased biased sample of clinicians, but I think they have excitement about the ability of polygenic scores to identify individuals of a very high risk. But I mean, at the same time, we have to realize that the sensitivity is very low. I think it's we know that from many studies that were published, including studies on embryos. We know that just identifying individuals at very high risk is going to find only a small proportion of the cases, and even the positive predictive value is going to be, it's not going to be very large. So we have to have that in mind, but still, it could be another tool, another way to stratify individuals by risk, and maybe give them better treatment. So it's early to say whether we will have it implemented at scale in Israel, but I think there is definitely a willingness to try it out and see how it works.
It sounds like Israel could be the other two that I think are promising are Estonia, weirdly, and Finland, because they're relatively small countries and they have good medical records and already some biobanking going on.
Yeah, but
Um,
they are a few years ahead of Israel,
I
unfortunately,
see. I see.
in terms of like setting up the biobank. We're just, I mean, again, like there is funding for, the funding was secured already a few years ago, but there were several problems that slowed down, if not, stopped altogether the this project, and only now we're starting more seriously. I mean, this is a project of the Ministry of Health. I'm a consultant there,
Mhm.
trying to help with experience on population genetics of Israel and so on.
I the example that stood out for me was breast cancer because people are already very familiar with BRCA, and the polygenic risk, can be as large for a fairly large chunk of women in the population compared to BRCA carriers. And so when you do very crude estimates of how much money you save by identifying, it's about an order of magnitude larger, the set of women who are as high risk as BRCA carriers, but only for polygenic reasons. And it looks like you can pay for, if you just take the cost savings that other people have published in other studies from early detection or early diagnosis, it looks like you can pay for genotyping all the women in a population just by breast cancer. So,
Yeah, and also genotyping is so cheap now that it's almost I mean, I don't think it's a major consideration anymore, like the cost of genotyping. I mean, it's
yep.
can be under $40 now, and if you do it at scale, it's even less. And also, if you genotype, you just need to genotype once for all diseases. So I don't think this is going to be the major consideration, but it's going to take a lot of effort and additional infrastructure and clinician time and so on to actually do the testing and interpret the results and provide genetic counseling and all that. And these, I think, these costs, I think, are going to be just much, much higher than the cost of genotyping. And this is where, it may end up not cost effective. But I think we can also learn, to do it efficiently. If we are convinced that the scores are sufficiently accurate.
Yeah, if you could convince the AMO that the HMO that they'll actually save money, then
Yeah,
maybe that'll switch people.
that always helps. Yes.
Yeah. Well, I appreciate your time. We went, it's now an hour and 20 minutes. So I apologize for taking up so much of your time,
No,
but I
it was
really
my
enjoyed
pleasure. Yeah.
the conversation. Yeah. I would love to come back and talk to you about Ashkenazi, the kind of history, genetic history. You know, d- one thing I wanted to ask you before we go is for Sephardic Jews, do you have similar results for the effectiveness of European trained predictors and things like that?
Um, so first of all, I would emphasize that what some people in Israel call Sephardic Jews. I mean, they're really, we have North African Jews, and Sephardic Jews meaning Spanish Jews, and then we have Middle Eastern Jews, mostly from Iran and Iraq. And there is so we know from genetic studies, from population genetic studies, we know that North African Jews are genetically relatively similar to Ashkenazi Jews. They are distinct, but quite close. So if I would have to guess, I would guess that the accuracy of polygenic scores will be somewhat lower, but not too much lower. But regarding Middle Eastern Jews, I think, I mean, it's really there's no data, and they are more genetically distinct from the other Jewish communities. We don't have a good clue. I mean, like, there is the only Middle Eastern data at scale that we have is coming from Qatar. Actually, just like a couple of days ago, they had a paper out on polygenic scores for cancer, but they didn't have enough cases to actually determine the accuracy of the score. So really, there's no data from the Middle East on the accuracy of polygenic scores. That's a pretty big gap in the literature
Mhm.
in this area.
Great. Well, I couldn't resist slipping in that last question. Shai, thank you very much for being on the podcast. I'm really happy that we did this, and I would love to have you back again if you have some more time.
Yeah, when I have new papers out, and we have we have more to discuss.
Yeah, that would be great.
Yeah, it was fun. Yeah, thank you for having me.